Hello friends, today's topic will be in the continuation with the tissue culture. So today I will be discussing about the haploid culture. Before I'll start with the haploid culture, थोड़ा सा brief कर लेते हैं कि haploid plants होते क्या हैं. See, the haploid plants, they bear a gametic chromosome number of a species, and they are generally derived from the gametophytic tissues that are developing during the reproductive phase of the plant. अब gametophytes कैसे आते हैं? Gametophytes develop after the meiosis. Both in anther to the process of microsporogenesis and in the ovule to the process of megasporogenesis. अगर हम बात करते हैं हैप्लॉइड माइक्रोस्पोर्स की, haploid microspores, they develop in pollen grains. जिसको हम मेल गैमेटोफाइट भी बोलते हैं, ठीक है. Whereas the haploid megaspores, they generate 8-celled embryo sac. जिसको क्या बोलते हैं? फीमेल गैमेटोफाइट. And this bears a egg cell. Right? So what happens? There is a fusion of haploid sperm cell from the male gametophyte with the haploid egg cell from the female gametophyte. And upon the fertilization, they will produce a zygote. And that will develop into an embryo, restoring the somatic chromosome number of the species. ठीक है. अब बात आती है an interruption of normal sexual development that can be either natural or induced. Causes the microspores or the megaspores to undergo mitotic division. Without fertilization. Eventually, that will result in a haploid plant. So such an extraordinary development of these gametophytic tissues can be induced in plant tissue culture, resulting in androgenesis or gynogenesis of the higher plant. So we will be discussing these processes. So first we will be discussing about the anther culture. See, this anther culture was first demonstrated by Guha and Maheshwari. And what they demonstrated is the possibility of androgenesis by the anther culture. So they used a plant which is Dhatura inoxia. See, although anther culture has been the most successful mean to obtain a haploid plant, microspores culture or ovule culture have been successful, especially for plants with anther culture has failed. So when we talk about the anther culture, this is called as this is called as androgenesis. And what is this? It is a technique. It is a technique by which the developing anther from unopened flower bud are cultured on a nutrient medium where the microspores within the cultured anther develop into callus tissues that give rise to haploid plantlets. इसको एंड्रोजेनिक हैप्लॉइड्स भी बोल देते हैं. So as we know that anthers are haploid, right? So they will be giving rise to the haploid plantlets. Now the haploid plants which develop from the anther culture either directly or indirectly through a callus phase, we will be discussing about the direct androgenesis. Okay. Direct androgenesis that mimics the zygotic embryogenesis However, there is neither a suspensor nor an endosperm is present. At the globular stage of a development, most of the embryos are released from the pollen cell wall, that is the exine. They continue to develop, and for after 4 to 8 weeks, the quadridense unfold and the plantlet merges from the anther. This is the direct way, right? So another is the indirect androgenesis. Indirect androgenesis, here the early cell division pattern is similar to that in the zygotic embryonic and the direct androgenic pathway. But after the globular stage, irregular after the globular stage, irregular and asynchronous division occurs, and callus is formed. So this callus must undergo organogenesis for the haploid plant to be recovered, right? So let us discuss about some applications of this anther culture. First is, we are doing it for the production of the haploid plants. Second is the production of homozygous diploid lines through chromosome doubling. So thus it will reduce the breeding cycle. Third application is the production of useful gametoclonal variations. So this production of haploid has been done. Approximately 250 species are species of the plants it has been done, and they are most common in the families like, it is done in the families like Solanaceae, Cruciferaceae, Gramineae, ठीक है, इन families में ये किया गया है. Now let us discuss about another culture, that is the ovule culture. Ovule culture, ovule क्या है? Ovule is a megasporangium covered by integuments. So ovule culture, या फिर इसको बोल देते हैं gynogenesis. It involves the development of haploid from unfertilized cells of embryo sac present in the ovule. So like as with the androgenesis, gynogenesis, haploids may develop directly or indirectly, okay, by the regeneration from the callus. So let us discuss about first the direct gynogenesis. Direct gynogenesis, it usually involves the egg cell synergids or antipodal cells with organized cell division leading first to the formation of pro-embryos and then to well differentiated embryos. This is for the direct one. Coming to the indirect gynogenesis. Callus may be formed directly from egg cell synergids, polar nuclei, or antipodal cells, or may develop from pro-embryos. So plants regenerated from the callus may be, so here the plants regenerated from the callus may be haploid, diploid, or mixobolite. So the relative scarcity of haploid cells within an ovule compared to the 1,000 generally found within the anther has made ovule culture or gynogenesis less attractive, okay, as compared to the anther or microspore culture for developing a haploid plants. However, for few species like onion, ovule culture has been successful, okay, and there are certain applications of the ovule culture. Applications. The first is, again, it's the use for the production of haploid plants. Then recovery of the hybrid embryos overcoming the embryo abortions at very early stage of development of the zygote due to incompatibility barriers. Next is for the achievement of in vitro fertilization. So I'll stop over here. Thank you so much. We will discuss somatic embryogenesis, organogenesis in the next class. Thank you.